Fatty acids. Any of the group of a long chain of hydrocarbon derived from the breakdown of fats. It has a single carboxylic group and a aliphatic tail. Fatty acid biosynthesis. 1. Diet provides abundant amounts of fatty acids. 2. Excess caloric intake of carbohydrate leads to storage as FAs. 3. Fatty acids are stored as triglycerides. 4. Liver and adipose tissue have particularly high rates. 5. But all tissues can synthesize fatty acids. Occurs in the cytosol. Requires 2N ADPH. Pyruvate Dehydrogenase. This enzyme converts pyruvate to acetyl-CoA *1*. Citrate Synthesis. This enzyme converts oxaloacetate to citrate, which then can be transported out of the mitochondria through the citrate transporter *2*. Citrate lyase. This enzyme converts citrate to oxaloacetate, consumes ATP and CoA, produces acetyl-CoA, which then can be used for FA synthesis in the cytoplasm *3*. Mallet Dehydrogenase. This enzyme reduces oxaloacetate to malate in the presence of high levels of citrate and pyruvate. Malate is not exported via malate transporter, consumes NADH *4*. Malic Enzyme. In the presence of high levels of citrate and pyruvate, this enzyme converts malate to pyruvate, which can be imported to the mitochondria via pyruvate transporter, produces NADPH *5*. Pyruvate carboxylase. This enzyme converts pyruvate back to oxaloacetate in order to restart the cycle of shipping acetyl-CoA from the mitochondria to the cytosol. Citrate and pyruvate transporters. These transporters are responsible for moving excess acetyl-CoA from the mitochondrial matrix to the cytosol to begin FA biosynthesis. Acetyl-CoA carboxylase. This enzyme carries out the first committed step of FA synthesis, the synthesis of malonyl-CoA *. 3 parts. 1. Biotin carboxylase. 2. Carboxylase transferase. 3. Biotin carrier protein that is loaded with biotin, input of *CO2 ATP. Biotin as cofactor, active only in a polymerized form, inactive in a monomeric form or phosphorylated polymerized form. This is the main site of FA regulation *. Fatty acid synthesis complex. Once the acetyl-CoA is in the cytosol and converted to malonyl-CoA, this enzyme complex makes the fatty acid. Acyl carrier protein. This component of the fatty acid synthesis complex is a small protein involved in fatty acid biosynthesis. It holds the growing fatty acid as it is being synthesized and releases it once it has reached its final length. It links a serine to a phosphopentathione, which is important for attaching the acetyl-CoA. In preparation for FA biosynthesis, acetyl-CoA is loaded onto this ketoacyl ACP synthesis. This component of the fatty acid synthesis complex has a cysteine, which is important for attaching the acetyl-CoA. Once acetyl-CoA is loaded onto the FASC, malonyl-CoA is loaded onto this enzyme *. Condensation. This is the first step of FA biosynthesis. It combines acetyl-CoA and malonyl-CoA to produce acidoacetyl-CoA. The methyl end of the molecule comes from the first acetyl-CoA incorporated. Loses CO2. The elongated FA chain now is on the shush group of ACP. Reduction. This is the second step of FA biosynthesis. The methyl end of acidoacetyl-CoA is reduced to N. Alcohol. NADPH is the reducing equivalent. Dehydration. This is the third step of FA biosynthesis. Reduction of double bond. This is the fourth step of FA biosynthesis. It again uses NADPH as the reducing equivalent. Translocation. This is the fifth step of FA biosynthesis. The butyl group is moved from the ACP shush group TOT. EK's shush group. FA biosynthesis continues by following this step by loading another malonyl-CoA onto the now open ACP, and around we go again. Thioesterase. In biosynthesis, when the FA chain reaches 16 carbons, palmitic acid is released by this enzyme. Chain elongation. Regulation of ACC. To regulate FA biosynthesis, substrates act on this important step. Citrate and PPP, high CARB diet, malonyl-CoA, palmitoyl-CoA, AMP kinase and pKa, high fat diet. AMP kinase. This enzyme will phosphorylate and inhibit ACC in FA synthesis and HMG-CoA reductase in cholesterol synthesis. Glucagon. Asterisk to gluconeogenesis to citrate to malonyl-CoA to carnitine acyl transferase 2. fatty acid oxidation to in ketone bodies, to CAMP to pKa2. Phosphorylation of ACC to ACC activity to malonyl-CoA to Fa import to mitochondria. Fatty acids. To malonyl-CoA to fatty acid transport to fatty acid oxidation. Insulin. To ACC to malonyl-CoA to fatty acid transport to fatty acid oxidation to fatty acid synthesis. Increase AMP. To AMP kinase to ACC activity to malonyl-CoA and fatty acid synthesis to fatty acid transport and fatty acid oxidation. Pentose cycle. Glucose 6-phosphate dehydrogenase. Number 1 This enzyme is highly regulated in liver and adipose tissue that is dependent on the NAD/NADPH ratio. High carbohydrate diet increases activity, high fat diet decreases activity. Insulin-induced transcription factors. 6-phosphogluconolactonase. Number 2 6-phosphogluconate dehydrogenase. Number 3 Transcutaneous. This enzyme of the pentose cycle is important for moving 2 carbon units. It includes TPP, which stabilizes the transfer. Transaldolase. This enzyme of the pentose cycle is important for moving 3 carbon units, no TPP because the 3C unit is transferred as a stable Schiff base. Xylose 5-phosphate. Fatty acid elongation. This process adds 2C to palmitate by using malonyl-CoA. Coenzyme A palmitate can be elongated, requires NADPH, occurs in her, produces STEARATE. Fatty acid desaturation. The body uses fatty acycl-CoA desiderases that introduce double bonds at positions 5, 6 and 9. The essential fatty acids are above 9 so can't be synthesized. Omega-6 linoleic acid parent omega-3 linolenic acid. Desiderases introduce double bonds at specific positions in a saturated fatty acid chain. Takes place in the ear. By membrane-bound enzymes, mammals can only incorporate a double bond between C9 and the carboxyl group, not beyond C9, X9, 6, 15. Palmitate. This is saturated fatty acid with 16C, no double bonds, important outcome from FA biosynthesis. Stearate. This is a saturated fatty acid with 18C, no double bonds, important outcome from FA biosynthesis. Carbohydrate. A polyhydroxyaldehyde or polyhydroxyketone, are a substance that gives these compounds on hydrolysis. Monosaccharide. A carbohydrate that cannot be hydrolyzed to a simpler carbohydrate. Monosaccharide formula. CnH2 non. Aldose. A monosaccharide containing an aldehyde group. Ketose. A monosaccharide containing a ketone group. Glyceraldehyde. Simplest aldose, contains one stereocenter and exists as a pair of enantiomers. Fischer projection. A two-dimensional representation for showing the configuration of tetrahedral stereocenters. Monosaccharide. The O on its penultimate carbon is on the right in a Fischer projection. L-monosaccharide. The O on its penultimate carbon is on the left in a Fischer projection. Anomeric carbon. The new carbon stereocent are created in forming the cyclic structure. Anomers. Stereoisomers that differ in configuration only at the anomeric carbon. Beta. Means that the O on the anomeric carbon is on the same side of the ring as the terminal CH2O. Alpha. Means that the O on the anomeric carbon is on the side of the ring opposite from the terminal CH2O. Pyranose. A six-membered hemiacetal ring. Furanose. A 5-membered hemiacetal ring. Mutarotation. The change in specific rotation that accompanies the equilibration of alpha and beta anomers in aqueous solution. Glycoside. A cyclic acetyl derived from a monosaccharide. Glycosidic bond. The bond from the anomeric carbon to the R group. Aldehyde. The reduction product. Sorbitol. Found in the plant world in many berries and in cherries, plum, pears, apples, seaweed and algae. Aldonic acid. Oxidation product. Reducing sugar. A carbohydrate that reacts with an oxidizing agent to form aldonic acid. Disaccharide. A carbohydrate containing 2 monosaccharide units joined by a glycosidic bond. Sucrose. Most abundant disaccharide in the biological world, it is obtained principally from the juice of sugarcane and sugar beets. This is a non-reducing sugar. Lactose. The principal sugar in milk makes up to about 5 to 8% of human milk and 4 to 6% of cow's milk. Consists of D-galactopyranose bonded by a beta-1,4 glycosidic bond to carbon-4 of D-glucopyranose, reducing sugar. Maltose. Present in malt, the juice of sprouted barley and other cereal grains, consists of 2 units of D-glucopyranose joined by an alpha-1,4 glycosidic bond, reducing sugar. Polysaccharides. A carbohydrate consisting of large numbers of monosaccharide units joined by glycosidic bonds. Starch. A polymer of D-glucose can be separated into amylose and amylopectin. Amylose. Composed of unbranched chains of up to 4,000 D-glucose units joined by alpha-1,4 glycosidic bonds. Amylopectin. Contains chains up to 10,000 D-glucose units also joined by alpha-1,4 glycosidic bonds. At branch points, new chains of 24 to 30 units are started by alpha-1,6 glycosidic bonds. Glycogen. Energy reserve carbohydrate for animals, is a branched polysaccharide of approx 1 million glucose units joined alpha-1,4 and alpha-1,6 glycosidic bonds. Cellulose. A linear polysaccharide of D-glucose units joined by beta-1,4 glycosidic bonds, insoluble in water. Acidic polysaccharides. A group of polysaccharides that contain carboxyl groups and/or sulfuric ester groups, and play important roles in the structure and function of connective tissues. Hyaluronic acid. Contains from 300–100,000 repeating units, most abundant in embryonic tissues and in specialized connective tissues such as synovial fluid. Heparin. Heterogeneous mixture of variably sulfonated polysaccharide chains. Carbohydrates. Sugars and starches, polyhydroxylaldehydes and ketones, are compounds that can be hydrolyzed to them. Carbohydrates are classified into three groups: mono, di, and poly. Carbohydrates are sugars, starches, polyhydroxyaldehydes, ketones, are compounds hydrolyzed into them. Monosaccharides. Simplest, 3 to 6 carbons, carbonyl group and O groups. Monosaccharides are blank compounds. Polar. Monosaccharides have blank melting points. High. Photosynthesis. Process that uses energy from the sun to convert carbon dioxide and water into glucose and oxygen. C1. Aldehydes. C2. Ketones. Simplest aldose. Glyceraldehyde. Simplest ketose. Dehydroxyacetone. Constitutional isomers. Glyceraldehyde and dehydroxyacetone both have molecular formula C3H6O3. Most prevalent aldose. Glucose. 3 carbons. Triose. 4 carbons. Tetrose. 5 carbons. Pentose. 6 carbons. Hexosa. Many polar functional groups equals. Hydrogen bonding. Hydrogen bonding equals. Very water soluble. Monosaccharides are very water soluble. All carbs, except for dihydroxyacetones, contain one or more chirality centers. Prefix L used. O used on left side. Prefix B used. O used on the right side. Naturally occurring enantiomer. D. Unnatural isomer. L prefix. Fischer projection formula. A carbon atom is located at the intersection of the two lines of the cross. Fischer projection formula. The horizontal bonds come forward on wedges. Fischer projection formula. The vertical bonds go back on dashed lines. What determines whether a monosaccharide is D or L? The configuration of the chirality center farthest from the carbonyl group. What are D sugars? glucose and all other naturally occurring sugars. D equals dextrose equals right. For amino acids, these are unable to form. Proteins. Glucose. Dextrose, blood sugar. Blood glucose levels 70 to 110 milligrams (DL). Excess glucose is converted to the polysaccharide glycogen. Insulin. A protein produced in the pancreas regulates blood glucose levels. When glucose concentration increases after eating, insulin stimulates the uptake of glucose in tissues and its conversion to glycogen. Diabetes. Produce insufficient insulin to adequately regulate blood glucose levels and the concentration of glucose rises. Galactose. Form disaccharide lactose, stereoisomer of glucose. Fructose. One of two monosaccharides that form disaccharide sucrose, found in honey. Catahexosa. Honey, 2x sweetness versus sucrose. Galactosemia. Lacks enzyme to metabolize galactose. Galactosemia accumulates in blood and causes cataracts and cirrhosis. Hydroxyl and carbonyl groups react to form a ring. The first step in cyclization is to rotate glucose 90 degrees. The second step in cyclization, the chain must be twisted around, forming a 6-membered ring. Cyclization yields 2 isomers. Rings are called Haworth projections. The alpha enomer, called alpha-D-glucose, has the O group on the anomeric carbon drawn down. The beta enomer, called beta-D-glucose, has the O group on the anomeric carbon drawn up. Haworth projections. Flat, 6-membered rings used to represent the cyclic hemiacetals of glucose or other sugars. Aldehyde group is oxidized to a carboxylic acid. Disaccarides. One monosaccharide hydroxyl group reacts with another hydroxyl group. Disaccharides glycosidic linkage joins the alpha and beta rings. Hydrolysis of maltose yields 2 glucose molecules. Alpha glycoside. Glycosidic linkage oriented down, below the plane of the ring that contains the acetyl joining the monosaccharide. Beta-glycoside. Glycosidic linkage oriented up, above the plane of the ring that contains the acetyl joining the monosaccharides. Hydrolysis enzyme of disaccharides. Amylase. Disaccharide in milk. 1 galactose ring and 1 glucose ring joined by 1 to 4 beta-glycosidic bond. Lactase. Cleaves 1 to 4 beta-glycosidic bond. Lactose. Principal disaccharide found in milk form both humans and cows. Lactose intolerant. Individuals who are lactose intolerant no longer produce the enzyme lactase. Lactose intolerance is prevalent in Asian and African populations whose diets have not traditionally included milk beyond infancy. Sucrose. The disaccharide found in sugar cane and the compound generally referred to as sugar is the most common disaccharide in nature. Sweet and low. Saccharin. Aspartame. Sold as equal, hydrolyzed into phenylalanine, cannot be processed by those individuals with phenylketonuria. Sucralose. Sold as Splenda, similar to structure to sucrose. Sucrose is sweet, many calories. Polysaccharides. Contain 3 or more monosaccharides joined together. 3 prevalent polysaccharides. Cellulose, starch, glycogen. Cellulose contains glucose rings joined in 1 to 4 beta-glycosidic linkages. Starch and glycogen contain glucose rings joined in 1 to 4 alpha-glycosidic linkages. Cellulose. Found in the cell wall of nearly all plants, where it gives support and rigidity to wood, plant stems and grass. Cellulose enzyme. No enzyme to hydrolyze. If there is no enzyme for digestion, no assimilation into blood and not available to cells. Starch. Polymer of repeating glucose. Starch is present in corn, rice, wheat and potatoes. First type of starch is amylose. Amylose. Unbranched polymer linked by 1 to 4 alpha-glycosidic linkages. Second type of starch is amylopectin. Two types of starch: amylose and amylopectin. Amylopectin, branched polymer linked 1 to 4 alpha and 1 to 6 alpha glycosidic linkages. Glycogen equals animals. Glycogen is stored in the liver and muscle cells. Glycogen is hydrolyzed to glucose by the liver. Glycogen is highly branched equals many hydrolysis sites. Both forms of starch are water-soluble. Both forms of starch are hydrolyzed to glucose with cleavage of the glycosidic bonds. Human digestive system has the necessary amylase enzymes to catalyze process. How many blood types are there? 4. Each blood type is based on 3 or 4 monosaccharides attached to a membrane protein. Each blood type has the monosaccharides galactose, fucose and N-acetyl-D-glucosamine. Type A blood contains monosaccharides galactose, fucose and N-acetyl-D-glucosamine. N-acetyl-D-galactosamine contains galactose, fucose and N-acetyl-D-glucosamine, and D-galactose. Type AB contains both type A and type B carbs. Type O, galactose, fucose and N-acetyl-D-glucosamine, universal donors because no antibodies to type O are produced to those with types A, B and AB blood. Type O are called universal donors. Type AB are called universal recipients.